Hello, future nurses and science students. We are fungi and we represent a completely different kingdom of microscopic organisms. If you recall our past discussions, bacteria are simple prokaryotes and viruses are non cellular outlaws. We, however, are eukaryotes. This means our cells look a lot like human cells. We have a distinct nucleus, a protective nuclear membrane, and complex organelles like mitochondria and an endoplasmic reticulum. In your nursing courses, especially when studying pharmacology and infection control, you will find that our advanced structure makes us a unique clinical challenge. Because we are so biologically similar to human cells, creating an antifungal drug that kills us without accidentally harming your patient's healthy cells is a delicate balancing act. Let's break down our structural blueprint so you can master your next exam. The two faces of fungi, yeast versus molds. Fungi exhibit two distinct morphological forms depending on the temperature and environment. Some fungi are even dimorphic, meaning they can switch between both shapes to survive. 1. Yeast, the single-celled spores. The structure: we are microscopic, single-celled, round or oval shapes. Reproduction: we don't divide cleanly down the middle like bacteria. Instead, we reproduce through a process called budding, where a tiny baby cell swells out from the parent cell, pinches off, and grows. Clinical example: Candida albicans, the opportunistic culprit behind oral thrush and vaginal yeast infections. 2. Molds, the multicellular networks. The structure: we are multicellular, growing in long thread-like filaments called hyphae, singular hypha. As these threads branch out and intertwine into a visible furry mat, that network is called a mycelium. Reproduction: we reproduce by releasing thousands of microscopic, lightweight spores into the air, which can be inhaled or land on surfaces. Clinical example: Aspergillus, which can invade the lungs of immunocompromised patients. The structural blueprint: chitin and ergosterol. This is the most critical concept for your nursing pharmacology and pathophysiology boards. To survive, we build two unique structural components that human cells do not possess. These components act as the prime targets for antifungal medications. 1. The chitin cell wall. Unlike human cells, which only have a flexible cell membrane, we wrap ourselves in a rigid outer cell wall. However, our wall isn't made of the peptidoglycan found in bacteria. It is constructed from a tough, fibrous sugar compound called chitin, the exact same material found in the hard outer shells of crabs and insects. 2. The ergosterol cell membrane. Just underneath our hard chitin wall sits our plasma membrane. Human cell membranes use cholesterol to stay stable and fluid. We, on the other hand, manufacture a unique plant-like sterol called ergosterol to maintain our membrane structure. Pharmacology connection. Landmark antifungal drugs like amphotericin B and fluconazole diflucan work specifically by disrupting or blocking the synthesis of ergosterol, poking holes in our membrane and causing our cellular guts to leak out. Image demonstrating the mechanism of action of azole antifungals, inhibiting ergosterol synthesis in the fungal cell membrane. Common fungal infections, mycoses. Fungal infections are medically referred to as mycoses. They are typically split into two clinical categories based on how deep they invade. 1. Superficial, cutaneous mycoses, the surface irritants. These fungi love keratin, the protein found in human skin, hair, and nails. They cause localized, itchy, but non-life-threatening infections. Tinea pedis, athlete's foot. Tinea corporis, ringworm, which isn't a worm at all, but a circular fungal rash. Candidiasis, when normal bacterial flora is wiped out by broad-spectrum antibiotics. Candida albicans overgrows, causing a thick white coating on the tongue called oral thrush, or an itchy vaginal yeast infection. 2. Systemic mycoses, the deep tissue crisis. These infections occur when fungal spores are inhaled deep into the lungs, allowing the fungus to enter the bloodstream and spread to internal organs like the brain. The targets: healthy individuals rarely get systemic mycoses. These are opportunistic infections that primarily strike immunocompromised patients, such as clients with advanced AIDS. oncology patients undergoing chemotherapy, or transplant recipients on heavy immunosuppressive drugs. Clinical examples: Cryptococcus neoformans causing fungal meningitis, and Pneumocystis girovetii, a unique fungus causing life-threatening pneumonia in AIDS patients. Critical nursing implications and clinical application. The antibiotic trap. As a student nurse, you must anticipate that anytime a provider prescribes a long, powerful course of broad-spectrum antibiotics to treat a bacterial infection, like pneumonia, the drug will accidentally massacre the patient's beneficial normal bacterial flora as well. Without those helpful bacteria around to compete for resources, opportunistic yeasts will rapidly overgrow, leading to secondary fungal thrush or skin breakdown. Always monitor your patient's oral cavity and skin folds. Antifungal toxicity alert. Because fungi are eukaryotes like humans, systemic antifungal drugs like amphotericin B carry a massive side effect profile. Amphotericin B is famously nicknamed amphoterrible on the nursing floor because it is highly nephrotoxic. When administering it, a nurse must continuously monitor the patient's serum, creatinine, and bone labs, track strict intake and output (INO), and pre-medicate the patient to prevent severe infusion reactions like rigors, chills, and fever. Please like, follow, and share.